are about to enter one of the most tightly guarded and dangerous places on Earth. Here, the final confrontation is taking place in a battle that has lasted centuries, a battle that has claimed as many as 100 million human lives. Now, it is the enemy's turn to die. Locked away here at the Centers for Disease Control in Atlanta and at a similar facility in Moscow are the only surviving smallpox viruses on Earth. Soon, these last smallpox viruses will be destroyed, the first time we have ever deliberately made another species extinct. But even as smallpox eradication symbolizes our victory over many of the great killers of the past, other diseases, including the world's newest and deadliest plague, AIDS, continue to defeat us. This film is a report from a war zone. Every minute of every day, we're all in a battle for survival. Our enemies are countless invisible microbes that seek food and shelter within our bodies. If they succeed, they can make us ill or even kill us. We only survive this unceasing attack because we're protected by a remarkable defense. At its heart is an early warning system that can spot and quickly identify an intruder. Then, an armed response is mounted that's custom designed to counterattack the invader. This defense, our immune system, has evolved through millions of years of pitched battles with our enemies. Unfortunately, our enemies have evolved too. They're constantly probing our defenses, seeking its weak points. And all too often, they succeed. One of the most stunning of our enemy's victories came at the end of what had already been years of unprecedented slaughter on the battlefields of the First World War. In October 1918, as the Allies were beginning their final offensive, a new enemy struck that was deadlier by far even than the carnage of trench warfare. In a few months, it killed twice as many as died in 4 years of man fighting against man. The new enemy was a strain of influenza. Before it ran its course, it killed some 20 million people around the world. Today, every winter, the flu returns. Every winter, there's a chance the flu virus will unleash another major pandemic. The virus travels from person to person in water droplets that linger in the air or on surfaces for hours. Even in normal years, the flu virus infects some 50 million people in the US alone and kills about 10,000 of them. Modern medicine's major weapon against the flu is made inside tens of millions of hen's eggs. Grown within each egg are inactive versions of the flu virus, from which will be manufactured the current flu vaccine. And this year, as every year, it's a race against time to produce 30 million doses.
We have a crisis atmosphere that develops for a period of about 6 months. We can make no mistakes. We must meet the urgency of the U.S. population needs and those in Europe. And our people are living and breathing flu from morning till night, well into the early spring.
Vaccines have been our principal weapon against major diseases for the last 50 years. But the idea of vaccination, and the name, goes back 200 years to an English country doctor named Edward Jenner. He noticed that milkmaids who caught a mild disease from cows called cowpox seemed protected against one of the main killer diseases of the time, smallpox. Jenner took fluid from cowpox blisters and used it to deliberately infect a young boy with the idea that it would protect him against smallpox. The cowpox virus is called vaccinia. Jenner's idea of vaccination was widely ridiculed, but today we know why it worked. When a microbe like the smallpox virus invades the body, it is met by an army of white blood cells. One whole regiment of cells is armed with feelers called antibodies. Each cell has its own unique antibody with which it gropes the intruder. Cell after cell tries for a fit. Eventually one comes along that's a perfect match, and the cell is triggered to multiply into billions of cells. These pour out the appropriate antibodies, which by binding to the viruses lead to their destruction. The system is marvelously versatile. No matter what the invader, eventually an antibody cell will come along that recognizes it. But there's a price for this versatility. It takes time for the right cell to happen by, time to multiply itself up. And during that time, the smallpox virus can be doing its deadly work. But in those cases where the immune system wins the race, the victim recovers and is now protected by a permanent platoon of cells that already possess the right antibody, ready to pounce should the same enemy reappear. It's this immunity to reinfection that lies behind vaccines. The vaccine used in the 1960s and 70s in a worldwide campaign against smallpox worked like Jenner's cowpox vaccine. It contained a harmless virus that looked like smallpox to the body's cells and prepared them for the real thing should it come along. By 1979, smallpox had been reduced from a deadly plague to a laboratory curiosity, and today is literally history. Smallpox killed by invading and overwhelming our bodies before the defense could be organized. The vaccine we used to beat it was essentially a tip-off that an attack could be expected, so the defenses were put on alert. While smallpox was swift and brutal, once our defenses were ready for it, it had no strategic options left. But the flu virus is more cunning and is still a potentially deadly threat. Landing in Atlanta is flu's latest recruit in its constant battle with our immune defenses. This little box contains a new strain of flu virus from China. Unlike smallpox, the flu virus is constantly changing, so our immune system is always being caught off guard. The flu vaccine that worked against last year's viruses may not work against this year's. The virus arriving at the Centers for Disease Control is the latest candidate for causing a major epidemic. And it's up to Nancy Cox, head of the CDC's Influenza branch, to outguess it. It's her job to recommend which virus strains to include in next year's vaccine. And there's only room for 3.
Outbreaks in Beijing occurring in some primary and middle schools, and they've got influenza B. The vaccine manufacturers actually have to have all 3 strains chosen and to them so that they can begin producing the vaccine by early spring. And we have to anticipate what's going to happen the following winter. This is really rather difficult, and it puts us under a lot of pressure doing as many tests on as many viruses as we can in a relatively short period of time.
Sure, this is a test that we did.
Every year, Cox and her colleagues have just a few weeks in the late fall and early winter to predict what the flu will do.
Really good possibilities as a vaccine candidate.
There are two basic families of flu virus, type A and type B, and this year it's type B that looks most threatening.
Brazil, China, we had 3, 4 more from China. All of these isolates, they're not well inhibited by our current vaccine strain in Panama.
Yeah. We're seeing a lot of influenza B viruses circulating at the present time. And this is causing us some concern because we're expecting, on the basis of this, that we might have an epidemic of influenza B this coming season.
The virus just flown in from China is currently Nancy Cox's biggest concern. It's a type B virus that has been causing severe cases of flu in the province of Guangdong. The big question is, how different is it from the B strain that was used in last year's flu vaccine called B Panama? To find out, antibodies that work against B Panama will be tested against the B Guangdong virus. This will be the first clue Cox needs to decide whether the existing vaccine could stop B Guangdong if the virus makes it to North America.
Well, what happened?
Well, take a look for yourself. I've run the new Guangdong virus against the Panama antibodies, and it's not reacting at all well.
The number of red dots shows how well the vaccine would work. The new Guangdong virus is the second row from the bottom.
Okay, well that indicates that this Guangdong virus is a new variant, and we'll have to keep an eye out for that one.
Right.
But exactly how has the virus changed so that it could sneak right past the immune defenses put in place by last year's vaccine? The Electron Microscope reveals the answer.
Oh, this is nice. I can see the spikes pretty well here. Using an electron microscope, we can begin to see the structure of the virus, and we can actually see the spike, the surface spike.
These spikes are what our antibodies latch onto. And here's the secret of the flu virus's quick change ability. Inside the virus are just 8 genes. The genes of most living things are carried by DNA, where the genetic instructions themselves are safely enfolded within the 2 intertwining strands of DNA's double helix. Only rarely do mutations change these instructions. But the flu virus carries its genes on a single-stranded relative of DNA called RNA, which mutates much more easily and often. From this apparent weakness comes the flu virus's strength. Because its genes keep changing, so do its spikes. And antibodies able to recognize the old spikes now don't fit nearly so well. It's almost as if the immune system has never met the flu virus before, with predictable results.
I started working on influenza when I was a graduate student. And I think that the thing that has kept my fascination through all these years is that although we know a great deal about it, we can't predict what's going to happen next with any great certainty. It's like a puzzle.
In a freezer in her laboratory, Nancy Cox keeps samples of all the puzzles from the past, all the different guises the flu virus has gone under for the last 50 years, including some that caused devastating epidemics. In most cases, it's the spike of the virus that, by changing its shape, has fooled our immune defenses. A computer reconstruction of how a spike is built from a protein chain shows the complexity of its structure. Even a tiny change can be enough to make it unrecognizable to an antibody. The colored regions on this view of a tip of a spike show all the places it has changed over the last 15 years. With time running out to decide on next year's vaccine, B. Guangdong is looking different enough from last year's B. Panama virus that Nancy Cox is facing a difficult decision. Should she switch to B. Guangdong? Her answer will depend on whether it looks as if the new strain is spreading from its Chinese origins. And it's already turned up in Taiwan, Cambodia, Australia, Chile, Argentina, and Brazil.
The fact that the B. Guangdong is spreading makes it an excellent candidate for next year's vaccine. But on the other hand, we have other viruses which are also a little different from the Panama vaccine strain. And we have to watch them and see if they're spreading as well. And we really work right down to the last moment, watching a complex picture in order to see which virus we believe is the most likely one to cause the next year's epidemic and get that in the vaccine in time to stop it.
In February 1993, Cox made her call, and on her decision may hang thousands of lives. She decided to stick with last year's B. Panama strain, gambling that while B. Guangdong is on its way, B. Panama still poses the greater threat. Only time will tell if she guessed right, and she could be catastrophically wrong. The flu virus that in 1918 killed 20 million in a worldwide pandemic wasn't just a subtle variation on previous viruses, but one that had altered beyond recognition. Another such drastic shift occurred in 1957, when the Asian flu swept the world. Hong Kong flu struck in 1968. Again, it seemed that the flu virus had completely reinvented itself.
It was in 1968, with the Hong Kong pandemic, the worldwide epidemic, that the scientific community realized what was happening. Until then, we thought that the virus just drifted along, changed slowly, and at some time it accumulated a lot more changes.
But the changes in the 1968 virus looked much greater than could be accounted for by the sort of subtle annual facelift the virus usually undergoes. The spikes looked almost alien.
There'd been reports of influenza in horses in some parts of the world, some serious diseases in chickens, some in pigs. So we set out on a worldwide hunt. What a great excuse to travel the world and find viruses.
What Webster was looking for was a virus that could be the source of those strange spikes on the 1968 Hong Kong flu virus. And in Canada, he found it, a virus that infects ducks and that carries in its genes the instructions for the Hong Kong spikes.
It's very interesting in wild ducks because the viruses cause no disease and they spread from duck to duck through the water. But you can't catch influenza virus from a duck by eating it, by handling it. So how do they transmit to humans? And do they transmit to humans? Indeed they do. The Asian pandemic had 2 of its 8 genes from wild ducks. The Hong Kong had 3 of its 8 genes from ducks. So we're very sure that the duck is involved in this picture. But studies show that the influenza virus in ducks doesn't multiply in humans. So how do they get together? We realize now that the pandemics, these worldwide epidemics, come out of China, Southeast Asia. All of the big pandemics have names associated with that area. So here we are, we have influenza viruses in ducks in Canada and pandemics coming out of China. How do we put these 2 pieces of information together?
The answer hinged on the traditional living arrangements in rural China. Both ducks and pigs share close quarters with farming families who work limited areas of land. The 3 species are constantly exposed to each other's germs, while pigs and humans can actually catch each other's diseases. The pig turned out to be the virus melting pot Webster was searching for. It's able to harbor both the human and the duck flu viruses. The genes of the two different viruses infecting the same animal at the same time can get mixed up, so that a human flu virus can now contain a duck flu virus gene and a duck virus spike. In an instant, a totally new flu virus has been born.
And the immune system doesn't have time to respond to that. The vaccine manufacturers don't have time to make a new vaccine. And so consequently, the virus spreads in people, in nations and around the world, and causes this pandemic.
It's the sudden emergence of such a pandemic strain of the flu virus that is Nancy Cox's greatest nightmare.
In our surveillance, we're looking not only for the new variants as they come along that are put into each year's vaccine, but we're also looking for a new pandemic strain that might emerge in humans and begin to spread. And we want to find that new pandemic strain in time so that we could actually make some vaccine and make plans to protect at least part of our population.
Unless the way people, ducks, and pigs live together in China changes, it's not a question of whether a new flu pandemic will happen, but simply when. Unlike smallpox, the flu virus has far too many faces to ever allow it to be eradicated by a vaccine. The best we can do is to stay one step ahead. 400 years ago, a disease that has so far defeated all attempts to devise a vaccine against it came to this Brazilian port along with the first European colonists. Today, it continues to devastate the lives of millions, not by constantly changing like the flu virus, but by slipping right through our immune defenses by pretending to be human. This graveyard in Bahia, Brazil, is the final resting place of many of the victims of the disease, the colonists who themselves unwittingly introduced it.
When the cocoa plantations were started by the Portuguese colonialists, obviously they needed a lot of extra help, and therefore they brought in many African slaves. And with the introduction of the African slaves came new diseases. Among those is schistosomiasis or bilharzia, a devastating parasitic disease which rapidly then spread through the colonial populations and throughout Brazil.
Worldwide, schistosomiasis affects some 200 million people, 8 to 10 million in Brazil alone. Don Harn has come here in an attempt to develop a vaccine against the disease. Working with him are his Harvard colleague Evan Secor and Brazilian biologist Mittermeier Rice. From here in Salvador, they will travel 5 hours inland to the town of Itacara. In the vast interior of Brazil's northeast, the heat is fierce and water is precious.
Oh, well, and you can see that they don't have...
Itacara is so dry that the stream that runs through town was dammed to create a lagoon. The lagoon is the town's most valued asset, yet it is also what brings schistosomiasis to its people.
Everywhere you look through here. Everywhere.
Like most human diseases carried by parasites, another species is host to the parasite for part of its life cycle. For schistosomiasis, this alternate host is a freshwater snail that thrives on human waste. Living within the snail and stimulated by sunlight to swim free are thousands of larvae of the schistosome parasite. These larvae, less than 1/8 of an inch long, swim through the water until they find exposed human skin. Once in the body, the parasites travel through the bloodstream to settle in the veins that lead off the intestines. There, they grow into inch-long adult worms. The larger female and the smaller male lie together, feeding off the blood in a permanent act of mating. And during their lifetime, which can be as long as 20 years, produce billions of eggs. It's these eggs, which inflame the liver, that cause the hugely distended abdomens of schistosomiasis victims. The eggs themselves, finding their way via human waste back into the water supply, hatch into yet another stage of the schistosome's life cycle. These larvae search out snails, and the cycle of infection is complete. In Itacara, the route by which the infection cycle is completed is only too obvious, and it suggests an equally obvious way of trying to control the disease.
If we improve the education and if we improve the sanitation here, give latrines and teach them that they should use the latrine, I think that the transmission will decrease a lot.
But as the research team explores the town and gets to know the people, so much of Itacara's life centers on the lagoon that education and sanitation won't themselves be enough to break the cycle of infection. For Don Harn, Itacara is a dramatic reminder of why he began his quest for a vaccine against schistosomiasis. The key moment came 10 years ago, when as a young scientist, he visited a Brazilian village not unlike Itacara, a village where the people had already been bombarded with education about the disease.
When I asked them about, do they know about schistosomiasis? Sure, we know all about it. And I looked in the water and geez, there were these snails all over the place. And I said, do you know about snails? And one woman completely bent down and picked one up and held it in my face and said, absolutely, here it is. And that point convinced me that something other than educational means were needed to eradicate schistosomiasis. And it was really a gelling point to say, no, I didn't make the wrong career decision in looking for a vaccine for schistosomiasis, that indeed it was going to take a long time, but that a vaccine was clearly a way to go.
9-year-old Rogerio's story graphically illustrates the need for a vaccine.
Tô com calor, então bora ali no rio tomar banho, daí ele tá assim. Então, bora lá, daí ele tá assim, não, então ele tá assim, então bora lá e acabar. Ele foi, acabou, fiquei continuando a tomar banho dentro, acabar, nós caçava de novo passarinho. Daí nós ia tomar banho no rio, foi dia nós pegou cistosa.
When Rogerio was first diagnosed, his liver contained more than 1 million schistosome eggs. Drugs have taken care of the eggs for now, but repeated reinfections will further debilitate and could eventually kill him. And here is the central mystery of schistosomiasis. Why doesn't Rogerio's immune system defend him against the worm?
Okay, Rogerio. You've got this large organism that's a foreign body inside of yours. And so basically your immune system should be attacking it and killing it, but it doesn't. The adult worms live right in your blood system where the immune system, all the immune system factors are right there to kill it. Yet the parasites may live from 3, 5, it's been reported as long as 20 years. Why is this? Why is it that a foreign body can live in an adult that amounts a perfectly normal immune response to anything else?
The answer seems to be that the worm has devised an ingenious way of making itself invisible to the body's immune system. Like all foreign invaders, the worm bears on its surface identity tags, antigens, which label it as not belonging. It's these antigens, like the spikes on the flu virus, that are recognized by the cells of the immune system including so-called T cells that both coordinate and take part in mass attacks on foreign labeled cells. But the worm's trick is to hide its telltale antigens behind molecules it steals from its host, so that the host T cells see the worm as human and ignore it. Don Harn's plan is to outsmart the schistosome by aiming a vaccine not at the adult worm, but at the schistosome larva, hoping to catch it as it enters the body, and before it's had a chance to don its human disguise. The strategy will only work if the villagers' immune cells are able to recognize the larva as foreign. To find out, the research team collects blood samples from some 300 townspeople.
Pretty secure?
Yeah, it stays.
Once the blood is collected, it's a race against time, because the crucial confrontation between the immune cells and the larvae can only take place back in the lab in Salvador. After a 5-hour journey, blood from the Itacara volunteers is about to meet antigens prepared from the schistosome larvae.
These are antigenic preparations that we brought from Boston frozen, and I'm just thawing them out in order to put them in the assay.
Meanwhile, Don Harn extracts immune cells from the blood samples.
I'm separating out the red cells from the white blood cells, and the white blood cells fraction contains the T cells that we're really interested in looking at, analyzing the T cell response. Or what we're really looking to find sort of an answer to is how do the T cells respond to certain of these candidate antigens.
The candidate antigens are 2 of many that the schistosome larva bears on its surface. Now the question is, will the villagers' T cells attempt to attack and destroy them?
We're ready to put the cells in the incubator, so we're going to begin the proliferation assay. And normally what we do in Boston is we have a CO2 incubator, and the CO2 helps maintain the right chemical balance for the cells while they're proliferating. We don't have a CO2 tank here for the incubator, so to generate the same, it turns out you can just simply add an Alka-Seltzer tablet to the water. It generates just what we want, keeps the cells happy, and then we put it in a sealed chamber, we lock it up, and everything works. We bake in the oven for about 5 days till they're done.
At the end of the 5-day test tube battle, the T cells of the people of Itacara responded powerfully to the larval antigens, suggesting that Harn's idea to make a vaccine using these antigens might indeed work to fend off the schistosome before it puts on its human mask. The experiment is promising, but before Rogerio and his friends in Itacara will benefit, there remains a very long step from the test tube to real life. In its battle with humankind, the schistosome worm has exploited to its advantage a key property of our immune defenses, the ability to discriminate between friendly and unfriendly forces.
My immune system is designed to tell the difference between what is me and what isn't, and to leave me alone. You can imagine the havoc that would result if the system went wrong and started recognizing parts of me as foreign. In fact, many diseases are the result of the immune system mistakenly recognizing friend as foe.
Don Sanders, a trombonist with the Boston Pops, began noticing symptoms of what would turn out to be multiple sclerosis about 10 years ago.
About that time, I started to have some trouble with balance, and I started to have trouble with my eyes. The doctor I first saw, he was able to give me a diagnosis of MS inside of a year. I felt like the only thing to do was just to, well, take one step at a time, see what happens, and everything will be okay. But
it wasn't that simple. You know, things got to be much more complicated, and they eventually got worse. And it was about that time that I first heard about David Hafler.
Multiple sclerosis is a progressive disease caused by the immune system's turning against and attacking the body's nervous system. Get back up here. That's about what you were last time. David Hafler and Howard Weiner, working at Boston's Brigham and Women's Hospital, have come up with an extraordinary idea for a vaccine against MS. A vaccine that could revolutionize the treatment both of multiple sclerosis and other so-called autoimmune diseases. Very good. The damage done to Don's brain as a result of his own immune system attacking it can be visualized with the help of a computer.
We can strip away the skin, and here we have the actual outside of the brain. We're now stripping away the brain, which we can rotate three-dimensionally. And by this computer reconstruction, we can actually follow the MS plaques. And we can see from this image this yellow area, which represents swelling and inflammation from T cells, which migrate into the brain. This is manifested in Don by difficulty with coordination and weakness in his legs.
In Don's brain, T cells are attacking a protein in the outer covering of his nerve cells.
Dad? Yeah. Here's the spinal fluid on patient. Spin it down.
David Hafler wants to know why the T cells of MS patients turn renegade and attack their own brains. He's recreated the confrontation between T cells and brain protein in a laboratory dish.
In this culture dish, we have two types of cells. T cells, which we isolated from the spinal tap that we performed about 2 weeks ago. We cloned the cells and grown them in culture. And we mixed them with a self-protein, myelin basic protein, which is attached to another cell.
When scanned by a laser, the T cells light up as they recognize and start attacking the nerve protein.
Now, in this culture, here, we also see T cells, which appear to be recognizing and attacking self-protein. The trouble is, these T cells are from my own blood, cultured also about 2 weeks ago. Why should there be T cells in my blood that recognize brain protein?
So the discomforting fact is that we all have T cells capable of attacking our brains. Why don't they? And why do they in MS patients? Hafler has discovered that only in MS patients are these renegade T cells actually found in the brain. He believes that in patients like Don, the cells have been accidentally activated, perhaps by a virus, so that they penetrate to the brain and attack it.
So Don, how are you doing?
I'm doing well. I'm feeling good.
Mm-hmm.
I feel like things are going at least as well as they were last time I was here.
That was about 6 months ago.
Don is being treated by powerful drugs that suppress his entire immune system in an attempt to hold the renegade T cells at bay. Hafler's goal is a more targeted treatment.
If it's true that activated T cells are causing multiple sclerosis, one wants to very specifically turn them off. And one wants to do it in a very specific way, not affecting other T cells, which you need to fight off foreign proteins, foreign viruses that may come into the system.
In wondering how to turn off just the renegade T cells, both Weiner and Hafler found themselves thinking about food and about why the immune system doesn't attack what we eat.
What is the immune system going to do with this turkey sandwich as the proteins leak through my gut into my circulation? Well, nature developed a very simple way of dealing with this. Nature turns off immune responses to proteins that one takes in the mouth and ingests. This is necessary to survive, because if you had immune responses to everything you ate, you'd be in very deep trouble. About a decade ago, both Howard and I independently had the idea of feeding myelin proteins to patients with multiple sclerosis to see if we could turn off the immune response.
They started with rats
feeding them with myelin basic protein, then giving them an injection that normally produces an MS-like paralysis.
And we're continuing to feed them. Let me show you these two groups. This is an animal that was fed a control protein and was injected, and you can see the animal basically has a paralysis and is unable to move the back legs. This is not dissimilar to the MS patient we saw. This is an animal that was injected in exactly the same way, but was fed the myelin basic protein, and there's clear protection from the paralysis. I think that this will have implications not only for multiple sclerosis, but for other diseases. And we've done studies in animal models, in arthritis models, in uveitis, which is a disease of the eye, and in certain diabetes models. And we basically have found that in feeding the appropriate proteins, we're able to suppress those diseases as well.
It sounds almost too good to be true, that autoimmune diseases like MS could be treated by having patients simply eat the molecules their immune systems have turned against. But early clinical trials suggest that some MS patients can indeed be helped by this approach. And for Don Sanders, there is again some reason for hope in his determined battle to beat the disease that is gradually destroying his brain.
It would be so easy for me just to lay down and throw up my hands and say, I can't do anything anymore. I give up. But I can't do that. I won't do that.
Don's immune system has chosen the wrong target. But even more deadly is a disease in which the immune system itself is the target. In the history of our species, there has never been a microbial enemy that has totally defeated our immune system. Even the big killers, like smallpox, didn't always kill, and those who survived an attack were immune. But today we're facing a new enemy. It's more cunning and so more deadly than any we've encountered before. As far as we know, it rarely, if ever, leaves survivors. And it kills, not because it sneaks through our defenses, but because its target is the defense system itself. Kevin Middleton has come from rural Virginia to Washington, D.C. to meet his brother Gavin, flying in from San Francisco. Kevin hopes to save Gavin's life.
When we're apart for so long, we get together, it's as if we hadn't even been apart at all. We know each other, we look the same to each other.
It's like looking in the mirror. I think we are mirror image twins.
The brothers are identical twins, but there's one big difference between them.
I guess so, they marked their territory. Gavin had called me and told me that he was HIV positive shortly after he had learned of it. I felt very sad for him and for myself, knowing that there's a possibility that very soon my brother could die. I don't want my brother to die, and I know that he could die at any time, and it makes me very sad because we are so close.
The idea of knowing that you could get sick at any time, that I might become ill at any time, really has made my life more exciting. I live each day knowing that I could become sick tomorrow and yet relishing in the day.
Gavin and Kevin's unusual situation has brought them to the National Institutes of Health to take part in a study that may not only prolong Gavin's life, but could help find a cure for AIDS.
think so. We better hurry up and get in.
Hi.
Hi.
Mr. Middleton?
Yes.
Hi, I'm Cliff Lane.
Hi, Dr. Lane.
Hi, how are you?
I'm great.
How was your trip in?
Oh, it was fantastic. Great flight.
Came in from California?
Yes.
Terrific.
Cliff Lane heads the NIH's identical twin study.
Shape and ready to go ahead with this protocol. Okay, what I'd like to do is take a listen to your lungs.
His plan is to boost Gavin's immune system with a transplant from his brother.
I'm just going to feel for lymph nodes in the neck. One of the advantages of being able to work with identical twins is that we can transplant the immune system without having to worry about rejection by the recipient. As with any type of organ transplant, as close a match as you can possibly get, the better off you'll be. By having identical twins, you have a perfect match, and this is a very unique setting and allows us to test the hypothesis of whether or not this transfer of immune system elements will be of benefit in the treatment of patients with AIDS.
What makes the AIDS virus so devastating is that it attacks a type of immune cell called a helper cell that orchestrates the whole immune response. Killing the helper cell cripples the entire immune system. The first hope of the twin study is that Kevin's healthy helper cells will reinvigorate Gavin's immune response, while the virus itself is held at bay with the aid of antiviral drugs.
The best case scenario of this study would be that you actually completely reconstitute the immune system of the recipient at the same time that you suppress the virus with antiretroviral agents, and that new, now potent, very energetic and vigorous immune system will keep that virus totally suppressed or suppressed for a very extended period of time in the recipient. If that happens, then we really have something.
What they're hoping for is something that has never been achieved since the AIDS epidemic began.
If we could just essentially cure one patient, that's sort of the key, the trail we need to find out what we can do for other patients as well. I mean, we have antivirals, we have everything else. It doesn't do what we want it to do. We think working with the immune system may really be the way to go, and we're hopeful with the identical twins that we'll have a chance to do that and then use that to help other people as well. So
just relax or talk or whatever. You just can't move your arms.
The first step in the experiment is to collect immune cells from Kevin's blood.
He's got good veins.
It runs in the family, the blood. Don't look.
Kevin's blood contains all the cells of his immune system, the cells that make antibodies, the T cells that attack cells that look foreign, and the helper cells that command and control the system, and that in Gavin are being destroyed. One of the great goals of AIDS research is to develop a vaccine to help fight off the HIV virus. One aim of this study is to help sort out which of the immune cells being collected from Kevin, the so-called lymphocytes, are most helpful in fighting the virus when given to Gavin. That information is critical in designing an effective vaccine.
Like everybody else did.
What we hope to learn by the studies of transfer of lymphocytes what elements of the immune system are important. If we can learn which elements of the immune system are important in fighting the AIDS virus, we can try and develop vaccines that will elicit those types of immune responses.
The idea behind the experiment may be simple, but the chance it could be successful attracts a crowd of very intrigued AIDS researchers.
No problems? Not a problem. I'm excited about it already.
So you're in the right crowd.
Previous patients in the study are being given a single transfusion of lymphocytes. This boosts the immune response briefly, but soon the donated cells too are attacked by the virus. So the plan is for Gavin to get repeated doses of his brother's cells.
How many cells will be infused into me?
Billions and billions of cells.
And my brother will come back 10 days before my infusion and give up his lymphocyte?
What we're doing in this study is we are going to be giving the lymphocytes every 6 weeks for 3 times, and the hope is that with each subsequent infusion, we'll see a further increase in the lymphocyte count, so that rather than getting an increase and a drop, we'll see an increase and then another increase and then hopefully even a third increase.
Are you optimistic that this will provide any answers for a cure to AIDS?
Yes, we are. Actually, that's the reason why we've proceeded to this point. You're talking about a formidable problem, but we are optimistic that this approach, under these very special circumstances, gives us the best possible chance of being effective.
That's great.
If the experiment does work, it could one day benefit even those newly diagnosed with HIV who don't have an identical twin to give them a transplant. The transplant donor could be themselves.
You take cells out of an HIV-infected individual early in the course of disease, eliminate any virus in it, store it, and then at an appropriate time when you can suppress the virus with drugs that we hope we'll have at that point to reinfuse the marrow into that individual. So it's important for the individuals who are undergoing the study, but the implications for others are potentially enormous.
Kevin, your body looks just like that guy over there. Go over there and pretend you're him.
All right.
For Kevin and Gavin, now the waiting begins. Ironically, the study requires that Kevin's immune system gets weaker before he's eligible to receive the boost of his brother's cells.
I'm very excited that finally something may be done that will provide answers. Even if they're small answers to the puzzle, it's thrilling to know that I'm a part of that and Kevin's a part of that. We feel important, that we are needed.
In Gavin's bloodstream, a deadly battle is being fought between the HIV virus and an immune system that is at once the target of the attack and the main defense against it.
Thank you very much for coming. I appreciate it.
You have a good flight, and we'll see you in 6 weeks.
Okay.
Bye, guys. Bye-bye.
Soon, Kevin's cells will be the newest recruits in this battle. Immune cells as yet unscarred and perhaps, just perhaps, capable of swaying the outcome. It will be one more battle in a war that has gone on since people and the microbes that make a living in our bodies evolved. But on the outcome of this battle, a lot more than one life hangs.
Tomorrow night on The Secret of Life, history is made when a 4-year-old girl receives the first gene transplant.
She has really come a long way.
But where will our new